I'm Elliot Meyerowitz from the Division of Biology and Biological Engineering at the California Institute of Technology. In the first two parts of my iBiology presentation, I talked about the phylotactic pattern of plants, why it's important agriculturally to be able to understand how to change it, and about methods that we developed of live imaging and computational modeling that have enabled us to understand and to change the phylotactic pattern. In this talk, I'm going to continue with the theme of phylotactic pattern and talk about a surprising set of discoveries that we made as we thought about our earlier computational model. And the summary of those discoveries is that we found out that the phylotactic pattern depends not only on chemical signaling, as I presented in part 2, but also on physical signaling between cells as they press and jostle against one another. And we find that this sort of physical signaling is not confined to phylotactic pattern, but may be an important principle by which we will understand the development of many different parts of plants. So you'll remember that we started with phylotactic pattern, and here we see the pattern of fruits around a stem once the flowers have matured in this spiral pattern where each successive primordium averages 130 or 140 degrees around from the previous one. And we explained this pattern by a computational and experimentally based model in which Auxin induces new primordia at the appropriate angle by being transported to the appropriate positions in the periphery of the shoot apical meristem through an Auxin transporter called pin 1. Local high auxin concentration causes new primordia, and it gets high locally by transport through pin 1 and also by diffusion, which resists the active concentration of the auxin, which is created by the auxin transporter. And the auxin efflux carrier, the pin 1 protein, is polarized in cells and it points from one cell towards the neighboring cell proportional to the auxin concentration in the neighboring cells. And these two rules, when put into a computational model, reproduced with great fidelity the patterns of auxin movement and auxin concentration change that occurred during phylotactic patterning in a real living plant. And by contemplating the computational model, we were able to understand some old and some new results regarding phylotactic pattern in an intuitive way. So we succeeded in producing a hypothesis that's been tested in computers and in plants for phylotactic pattern. But there's a problem. Now remember, our computational model demonstrates in great detail exactly the types of auxin flux that we see in a real shoot apical meristem, and it does it because the model follows those two rules. I presented them as 3 rules in part 2, but 2 observations here in part 3. But one of them is very curious, and you should stop and think. This is no problem. Local high auxin concentration causes the new primordia. That's well demonstrated. And it gets high locally by transport and diffusion, also well demonstrated. But our rule that the auxin efflux carrier is polarized in cells and points toward neighboring cells proportional to their auxin concentration raises a problem. How does a cell know the auxin concentration of its neighbors? Each cell has in its nucleus a receptor that is well characterized in a number of different laboratories in plant biology to which auxin binds, causing the degradation of a set of anti-transcription factors, which then unleashes transcription in the nucleus and changes in the patterns of genes that are expressed in the cell. So a cell can read its own auxin concentration because it has an auxin receptor inside itself that responds differently to different concentrations of auxin. But how does a cell know the concentration of its next door neighbor? These cells don't read minds, they don't have ESP. So what is it that the neighbor is signaling to a cell that enables that cell to know the neighbor's auxin status as well as its own status? It can't be its own auxin receptor, because that auxin receptor is reading its own auxin concentration and the neighbors have a different concentration. So what's going on? We can think of a variety of models that might explain how one cell signals its auxin concentration to its neighbors. One is that auxin itself might be a signal, so that a cell exudes auxin through its PIN1 transporter, and somehow the cell next door picks up the gradient of auxin between the delivering cell, which could be high, and the receiving cell, which is low. Except that when we make computational models of this, they don't work. The cell wall is so thin and auxin diffuses so quickly that there's no practical way to get a gradient of auxin from one cell to another. So even if there were a hypothetical cell surface receptor that's a different receptor for auxin, it wouldn't know which cell the auxin is coming from and which cell it's going to, because you can't form a gradient over so short of a distance in a small molecule that diffuses as rapidly as does auxin. Could auxin induce a different diffusible substance that diffuses more slowly? That's a possibility. Could auxin activate a gene that makes a cell surface ligand that sticks out of one cell that's perceived by receptors that stick out of the neighboring cell, like ephrins in mammalian cells, which act that way? That's a possibility. But there's one other possibility that we had to consider that's a little bit of a radical possibility, which is that auxin causes cell expansion, as we discussed in the earlier parts, and as a result of that, it creates physical forces within the tissue that must affect neighboring cells. Let's think a little bit about what could be going on here. Here's a view, one of our time-lapse movies, showing the nuclei of shoot apical meristem cells stained with a fluorescent probe over a short time course of some hours, and we can see the nuclei where cells divide, they pop into two nuclei. And if we use our computer image processing methods to find the center of each nucleus, we come up with a picture like this, in which we've excluded the nuclei of cells that divide, and so we're using all the other nuclei as fiducial points for positions in the tissue. And so what we're seeing here is the effects of both the expansion of cells in the meristem and division of cells as they force the nuclei of undivided cells farther and farther apart. And if we analyze this computer representation with another stage of image processing, we can make a heat map of the expanding regions in the shoot apical meristem so that the regions where the nuclei are moving apart from each other more rapidly are orange and more slowly are blue. And we can make that in 3 dimensions. And what we see with these rectangles representing the principal directions of growth and the heat map, the colors orange, the most rapidly expanding cells, and blue, the cells that are expanding less rapidly. And what we see is the orange pattern fits exactly the pattern of auxin that we saw in our earlier auxin reporter slide in the previous talk. So that each place in the meristem where auxin is at higher concentration, the tissue is growing more rapidly, and at the places where auxin is at lower concentration, those places that won't soon be making a new flower primordium, the tissue is growing more slowly. And in fact, the time course of this image is such that very few of the cells divided, so what we're really seeing here is the direct effect of auxin causing the cells to expand in the positions that later will be occupied by the primordia of new flowers. So we know that the positions where new flowers are about to form and those cells that are high in auxin are pressing on their neighbors. And think about a plant tissue. It's quite solid. High turgor pressure, as we discussed in the last part, restrained by a cell wall, and it makes a tissue like a carrot. You crack it, and you hear that crack. That's the explosive decompression of the cells relieving the turgor pressure. They're pressing on each other like bricks in a brick wall press on each other. And imagine what would happen in a brick wall if one of the bricks started to expand and the others didn't. There'd be huge stresses in the surrounding bricks, and they would crack and they would fall out of the wall. So why doesn't that happen in this case, where we have the same degree of expansion, something that's very tight? And it's because the cells have, in some way, to respond to the stress adaptively. Now, why would we consider that stress to be a signal of auxin concentration that can be perceived by the neighboring cells? The model would be that as a cell expands, it stresses the common wall it has with a neighbor, and the plasma membrane of that neighboring cell then gets loaded with more PIN1, which causes the auxin to be directed towards the high auxin cell. So the cell has to be able to sense the stress on the common wall and then add more pin-1 to its plasma membrane, which is done by exocytosis. We know how the pin-1 gets there. And so that would be our model. Does this make any sense in terms of what we know about plants? Well, yes, it does, and it explains some earlier experiments, if true, that otherwise seem quite puzzling. I mentioned in the previous talks that if we add a drop of auxin to a shoot apical meristem, we can induce the formation of a new flower at that position in the meristem. But there's something else that you can drop in a new meristem and equally induce the formation of a new leaf or a new flower, and that's expansin, an enzyme that causes the weakening of cell walls directly and not through changing gene activity the way auxin does. And Fleming and collaborators over years starting in 1997 did a series of experiments where they showed that expansin acts as does auxin. But all the expansin is doing is weakening the cell walls and letting the cells expand. And somehow this triggers the flower development just as well as auxin does with all of its subsequent changes in gene activity. Similarly, pectin methyl esterase, which is an enzyme that affects cell wall stiffness, changes the phylotactic pattern when it's put on top of a shoot apical meristem. And so the target of auxin in inducing a new flower seems as likely to be the cell wall as to be the gene regulatory changes that auxin causes in an individual cell, so that we know that the cell wall is signaling somehow to the cells in the shoot apical meristem. Could it be that this signal is the one to which cells are responding so that they partition their pin-1 in the plasma membrane? So to see if this is a possibility, we made a computational model just like our earlier phyllotaxis models, in which the stress felt in walls between two cells reads out to pin-1 concentration in the lower auxin of those cells, and it creates a pattern, just like the phylotactic pattern we saw before, though locked up in this case because we didn't happen to put cell division in this model. And then what else we did in this model, which is a physical model of the shoot apical meristem in which the stresses in each cell are part of the computational modeling, we ablated a cell right in the middle of the meristem. And when we ablate that cell, the image on the left shows auxin concentration, and the image on the right shows where pin-1 is expected to go if we take a laser and we blow up a cell in a shoot apical meristem. So we did that. This is the expectation of the physical model, is that if you blow up a cell, you should cause added stress on the walls behind the cell that's blown up. This one's blown up, and then more stress on these walls and then subsequently on these walls as we move back. The reason for that is that the epidermis of a shoot apical meristem is as a whole under tension. It's being stretched. And this has been known for almost 200 years, that the epidermal layer of plants is under tension, resisting the turgor pressure and expansion of the cells underneath. One of the early demonstrations of this by von Sachs in 1880 was that he cut a dandelion stem right up the middle, just with a razor, and the two sides roll up like that because the epidermis is under tension, and that tension is resisting the pressing of the cells underneath, and that's equally true in the shoot apical meristem. So we have a stretched tissue, we eliminate a cell and eliminate its continued repair of its wall, so the wall weakens, the hole we've made enlarges, and that creates circumferential stress here and then here and then here as we move back. So the expectation of our stress model is that if we take a laser, a high-powered dye-coupled laser, and add it to our confocal microscope, and while we're watching, we kill a cell, and then we look to see what happens to the pin-1 in the cells surrounding the dead cell. If our stress model is correct, we expect to see the pin-1 move to the back of the cells surrounding the ablated cell, then to the back of the next cells and the back of the next cells, which is exactly what we do see. Where red here is pin-1. Those two blue things are the two cells that we killed, stained with a vital dye, and the pin-1 is moved to the back of the cells and then the back of these cells, and it's in the process of moving to the back of yet another layer of cells. So it behaves exactly as predicted by a stress model, but not at all the way you would predict if auxin or some other diffusible substance was creating the pattern, because this pattern results from physical changes and it's not the pattern you'd expect from chemical changes. Now you could ask if it's possible that there's some other chemical that's fooling us, that we've wounded a cell and it's exuding some sort of wound signal that makes the pin-1 go to the other sides. And we've made a number of control experiments to show that isn't it. And here's an example of one where we wounded the cell, just the cells of a shoot apical meristem, by scraping it with a needle. And if you do that, the pin 1 normally will go to the back of the cells next to the scrape and then to the back of the next cells, just according to the physical model. But if we relieve the turgor pressure on the right-hand side here, and we, by adding a non-metabolizable sugar to the medium that's equal in amount to the solutes inside the cells, and then we do the wounding, the pin 1 doesn't do a thing. So that there's not a chemical signal, it's a physical signal that's causing the realignment of the pin 1. And so what can we say about this then? We can make a model where auxin transport that we discussed in the last talk adds to the auxin concentration in a particular cell, causing that cell to grow. And a cell with more auxin grows more, which changes the tissue shape because it's pressing out on its neighbors, thereby creating a mechanical stress which feeds back to the pattern of auxin transport, so that we have a feedback loop whereby auxin controls its own flow by weakening the walls of the cells and causing cell expansion through a stress feedback loop. There's another element to this that we discovered while we were studying the pin 1 transport. So that since the epidermis of a shoot apical meristem is under tension, we can treat it as a pressure vessel. In the centuries before this one, engineers who built boilers had to understand the stresses in boilers to prevent industrial accidents, or at least to aim them towards the ceiling instead of the wall. And what they found was that spherical or hemispherical pressure vessels, boilers, have isotropic stresses. The stresses are the same in all directions through those, so they don't have any preferred direction in which they explode. But a cylinder has a preferred stress direction circumferentially that's twice as much as up and down, which is why an unreinforced cylinder of that type, when it gets too much pressure in it, will blow open with a long hole this way, because the stress is more in this direction than it is in this direction. We applied those same sort of pressure vessel calculations to our shoot apical meristem, which is an epidermal layer under tension, as is a boiler, so that we could estimate what some of the forces were in the course of making our pin 1 models. And what we saw was this, is that in the hemispherical part of the shoot apical meristem, at the very top, the stresses are the same in all directions, or isotropic. But in places like the boundaries between a flower primordium and the meristem, the stresses are very strongly tensional across the saddle point, and they can even be compressional in the orthogonal direction across the saddle point. And so that we have a pattern of stresses that results from the shape of the tissue and that's intrinsic to the shape of the tissue because the epidermis is under tension, and that can be calculated the same way boiler pressures are calculated. And so when we look at these tissues, we look here at one of the elements of the plant cytoskeleton, the microtubules, and we see the microtubules in the shoot apical meristem. We see the surprising fact that they line exactly according to our stress model. So that if we look at the microtubules in the central part here, they're in no particular preferred orientation. And these microtubules are what in plants are called cortical microtubules. They wrap around the cells in helical fashion just under the plasma membrane. It's these cortical microtubules that were first led to the discovery of microtubules in plants, which were the first discovery. When we look over at these boundary regions where the stresses are thought to be strongly tensional in this direction, the microtubules are entirely aligned with that. If we look over time at this, these microtubules in the isotropic regions continually change their direction, while these microtubules are fixed. And so we have a second result of stress in the shoot apical meristem, one that enables us to read out stress independently of pin 1, and we can see the correlation between microtubule patterns and pin 1 patterns, which helps to support the physical model of pin 1 localization. But there's another implication of this cytoskeletal aspect of stress that's very important for understanding the phylotactic pattern in the shoot apical meristem. So that the microtubules are aligned in parallel to the maximal principal direction of stress when stress is isotropic. It's a mouthful, but that's the hypothesis, and that's what the evidence supports. In fact, we can test that by compressing the meristem. We built a little vise and squeezed the meristem. As you can see here, we've started, then we squeezed it, and we kept the squeeze on for 6 hours, and the meristem keeps compressing. And as we do that, the microtubules at the top of the meristem stop continually realigning and become aligned, and they become aligned parallel to the principal direction of the tensional stress, which we can see here in this before and after picture. So on the left, we're squeezing before, after, and you can see the alignment of the microtubules is following the imposed pattern of stress that we've added. So again, experimental evidence that we can intervene with stress, or in another way, we can ablate cells with and change the stress pattern, as we did for pin 1, and cause a realignment of the cytoskeleton. So here's a physical model. It's called a finite element method model, a type of physical modeling of cells all under turgor pressure in a layer. Underneath there's another layer, and the layer underneath is pushing up on this one, so that this one is under, the layer we see on top here is under tension, just like in a real meristem. And the little white lines, difficult to see, but they're the principal directions of maximal stress in each of the cell walls, and they're quite at random at the top because the pressures, the stresses are isotropic in this hemispherical part of the meristem. If we ablate a cell in the computer, the stress is realigned to become circumferential to the ablated cell. Remember, that would make the pin 1 go back to these walls because these walls become more stressed, but the overall stress pattern is circumferential and around the hole that we create, so that if we do a live imaging experiment and we watch the microtubules when we ablate a cell, what happens is the microtubules realign within about 6 hours to being circumferential to the ablated cell. So just like for pin 1, which in 2 to 6 hours reorients to be the plasma membrane adjacent to the most stressed wall, the cytoskeleton of these cells realigns to be parallel to the principal maximal direction of stress when the stress is anisotropic. What does this mean for the cells? Well, here's some shoot apical meristem cells so that you can see what real cells look like in an electron micrograph. The nuclei are here. These dark things are nucleoli within the nucleus, which is here. And then we see the cell wall, about 80 or 100 nanometers thick, that's resisting all of this turgor pressure here. Now, the resistance to turgor pressure in a plant cell wall is due largely to the deposition of cellulose fibrils in the wall by cellulose synthase enzymes, which ride along the microtubules as tracks and deposit cellulose outside the cell through the plasma membrane in the direction of the microtubules in these cells. Individual cellulose fibrils, which are made up of numerous cellulose molecules twisted around each other like a thread, have the tensile strength that's about half that of steel. They're very strong and they strain very little under stress. And so they reinforce the cell in whatever direction they're laid down. If we look here, the green on this left-hand side, these are some cells on leaves that we've pictured, and the big green spots are Golgi vesicles depositing the cellulose synthase complexes near the surface, and the little green dots, if you look closely, are those complexes moving along the microtubules and laying the cellulose fibrils down. So what happens then? When we look at this, we have microtubule orientation responds to stress, and the microtubules align to the stress direction. The cellulose synthases ride along the microtubules and reinforce the cells in that direction, so that when the cells expand subsequently, they expand orthogonally to the cellulose fibrils at right angles to the direction in which they're laid down, because they resist expansion. So the direction of expansion of the cells in the shoot apical meristem depends on cellulose reinforcement, which depended on microtubule alignment, which depended on the stress pattern. So if you pull on a cell, after a few hours, its cellulose is lined up in the direction of the stress you created, and the cell starts to expand in the other direction. So that we have two different pathways that feed into the expansion of cells in the shoot apical meristem. One is the Auxin pathway, whereby auxin causes the cell to expand, and its rate of expansion is proportional to the auxin concentration, but the direction in which it expands depends on the microtubule orientation. So we have two stresses, one acting positively on cell expansion. An auxin-rich cell attracts more auxin because of the stress it makes and expands more, but one that resists that expansion because a cell that realigns its microtubules to the direction of expansion restricts expansion in that direction and causes expansion to occur in another. So it's a negative and directional feedback on cell expansion. Each of these affects the mechanical stress in the tissue, and each of these affects the other pathway. So we have overall a set of feedbacks through mechanical stress that controls the flow of auxin through the tissue, that controls the rate at which the cells expand in the tissue, and controls, therefore, which cells attract auxin and which don't, and so that expansion of the cells can trigger any pathway that auxin triggers, because expansion makes those cells attract auxin from their neighbors by a purely physical signaling, which then turns into a chemical signal, which then turns into a new flower. So what do we have? We've got a model now that's computationally supported, it's experimentally supported, but just like our earlier model, this model opens a bunch of new questions for scientific analysis. What are the sensors in each cell that allow the cells to sense in the cell wall stress, or that allow the microtubules to sense stress? How do they sense directional stress? They can't be molecules that are unoriented, because that wouldn't tell you the orientation of the stress. How do the microtubules respond to stress? How does pin 1 redistribute in response to the stress? Is this true only in the shoot apical meristem, or does the whole plant work this way? And can we model this entire system in our computer and get fully realistic developmental outputs from the combination of chemical and physical signaling that results from not only the gene activity patterns in the tissue, but from the physical state of the tissue, including its cytoskeleton and its cell walls? And just to give you an indication of a few of the directions we've taken these experiments, what we're seeing here is the microtubule pattern on the surface of a cotyledon, an embryo leaf. And you can see that the microtubules realign in quite a lively fashion, just as they do in the shoot apical meristem. These are the stomata, which have a particular stress pattern of expansion of a donut-shaped thing that causes the microtubules always to be aligned around the principal stress direction. If we look in detail at the microtubule pattern in these cells, we see that it changes quite a lot in some parts of the cells, but in other parts of the cells it's constant. And the parts of the cells where it's constant are those parts of the cells where the stress is highest, according to a physical model of the cell, and the stresses are created, in this case, by the shape of the cell, which is an oddly shaped cell, and which has a series of lobes and necks, and where the necks are is where the stress is highest and where the stress is most directional, where it's anisotropic and directional, and the microtubules are aligned and locked in those regions of these cells, and they vary in the other regions of the cells. So just like in the shoot apical meristem, the cells of the leaf have a cytoskeleton that responds to stress, and we learn something new, because the meristem cells are very small and they have a single direction of microtubules. These are large cells with a complex internal stress pattern resulting partly from the tissue they're in, but partly from the shape of the cells, which isn't the case of the cuboidal cells in the shoot apical meristem. And so we can see that the response of the microtubules is subcellular. If we have high stresses in one part of the cell, the microtubules will align. And if we have isotropic stresses in another part of the cell, the microtubules keep changing in their lively fashion. And so it's an indicator of mechanism that tells us that in the case of the microtubules, it's something local within the cells that's sensing stress and not the entire wall, while for pin 1 it seems to be something in the wall and it does not have to be local. We can do the same sort of ablation experiments with these cells as we did before and see alignment of the cells around the expanding hole that our laser ablation has caused. And we can even take mutations that affect microtubule behavior to see exactly which aspects of the microtubules are necessary for alignment, such as in this case, where you can see that there's no particular alignment of the microtubules in these cells. And the reason for that is that these cells are mutant in an enzyme that causes microtubules to be cleaved when they cross each other. So if you have crossing microtubules, one of them gets cut by this enzyme catanin, and this is a mutant in one of the 2 catanin genes in the Arabidopsis genome, so that it's that cleavage that causes the microtubules to align. How might that cause them to align to stress? There are many possibilities. The simplest might be that a stressed microtubule strains a little bit, and so it becomes less of a good substrate for the catanin, and so that the stress microtubules persist and the others get cut, which would eventually lead to a net alignment to cellulose biosynthesis and a subsequent direction of expansion of the cell in a particular direction. So we're on the case. We're trying to understand the subcellular and molecular mechanisms for these stress sensations, stress sensing mechanisms that lead to PIN1 relocalization and cytoskeletal reorganization. And we can see that they're of critical importance in any complete model of how a plant grows and develops. We should also ask ourselves whether such stress considerations might teach us a few things about the development of animals. And I think the answer to that is yes. You'll have to leave that to an animal developmental biologist and to another eye biology talk. So let's summarize. We started at the beginning of this talk with a standard textbook view of developmental biology, where gene action causes changes in cell behavior. Among those cell behaviors is cell-cell communication in a tissue, and that cell-cell communication feeds back to gene action. And this loop creates morphology through other cell behaviors. We have to add to this that the morphology itself feeds back on gene action because the stresses in the tissue which result from the shape of the tissue regulate auxin transport, which changes gene action, that the morphology of the cells directly affect the cell behavior, as we've shown with the cytoskeletal reorganization in response to stress, and the morphology of the cells, both their shape and the shape of the tissue in which they reside, therefore affects cell-cell communication, both by changing the stress pattern, because it changes the direction of expansion of the cells, and by changing the PIN1 direction in each cell, which therefore changes the way in which they communicate with their neighbors. So we go from the old-fashioned view of development as something that's played out in a genetic hierarchy to something where there's an interplay between the physical state of the cells and the physical state of whole tissues, the shape of the cells and the tissues, and the rates of expansion of the cells in the tissue that themselves control gene action and cell behavior, which in turn control the stresses in the tissue. So we have a series of new feedback loops that we have to consider in any model of plant development and probably of animal development, and it gives us a better possibility for predictive models in the future because we now understand what are some of the players and that physical stresses in tissues are as important as chemical signals in tissues for the formation of developmental pattern. As in part 2, I show again many of the people in my lab now and names of the people who contributed to this great collaborative project, and with great thanks, in this case, to the Department of Energy, which has funded all of our mechanics work, to the Gatsby Foundation in England, which has funded some of this work in the most wonderful way, and to the Gordon and Betty Moore Foundation, the Howard Hughes Medical Institute, and last but not least, either financially or in terms of their contributions, the National Institutes of Health, all of whom have funded my laboratory generously over the past years and enabled us to do this work.